All right, what we're going to talk about today is cellular respiration and cellular energy. And when we talked about cell transport, the term ATP kept coming up. ATP is the cash currency for any activity that your cell needs to do. So, whether or not you're a muscle cell and it's allowing me to talk right now by moving the muscles in my jaw, or the muscles in your core that are allowing you to sit upright as you're watching this, all of that is utilizing ATP. So, for your cells to function, for active transport, for muscles, for bioluminescence, which is the ability to glow, you have to have ATP. There is a couple of other kind of sources of quick energy that your cell can't use except for to make ATP or to make a longer term storage, which is glucose. So we store energy by making bonds and we release energy by breaking bonds. Well, the two molecules that we're going to mostly talk about are NADP, which is empty. And when we add energy to it, the bond that we make is the bond between a phosphate, that P, and a hydrogen, H, to make NADPH. So, in between the P and the H is a bond. When we make that bond, we store some energy. When we pop that hydrogen off and we break the bond, we release some energy. NADPH is a temporary energy storage unit that shuttles that hydrogen, and we consider the hydrogen and the bond that's associated with it our energy. It shuttles that to the next place where we can make ATP or we can make glucose, which is a sugar. The second and the best way, like I said, our currency of movement, of activity is ATP. It can be used to fuel your cell processes, making your muscles move, or can be put into long term storage as glucose. Both these chemicals are used in light independent reaction and in cellular respiration. And one of them is found in the making and the breaking of the sugar glucose. So, what is ATP? Let's go a little more in depth on this. It is a chemical used to store energy by plants and animals, and it's made of a 5 carbon sugar and 3 phosphates. The empty molecule, and I'm just going to go ahead to the next slide. The empty molecule is our sugar complex right here and 1 phosphate. Then, if I add a little bit of energy, I add a second phosphate and I make ADP. D standing for di or 2 phosphates. I add a little more energy and I create this bond. And now I've got 3 phosphates or ATP. ATP is a fully charged molecule. AMP is 50% charged and AMP is 0% charged. So again, we're going to use this ATP to fuel the muscles in your body. We're going to do membrane pumps like we saw in cell transport. We're going to make proteins with it. And some organisms have the ability to glow. So the gene that's making this jellyfish glow, they've actually taken that gene out of the jellyfish, put it into a mouse, and the mouse glows. Not great for the mouse because then at night he can't hide and the hawks and the owls see him and he gets eaten. But we can make things glow. Well, why would things glow in nature? So you have things like fireflies, if you've lived somewhere else in the country and their butts glow, and they do that so that they talk and they pulse their butts in different language, essentially. We've got things like the lanternfish from Finding Nemo that's got the little dangly bit that has a glowing ball of symbiotic bacteria in here, and it glows, and the other fish go, ooh, pretty light, and they swim it forward towards the glowing light, and the anglerfish eats them. So, how are we going to use that stored energy? How are we going to use the energy that you ate with breakfast or that you ate with lunch? We're going to do that through a process of cellular respiration. So, cellular respiration is the process of releasing energy that is stored as glucose, as sugar. We can do it by heterotrophs or autotrophs. Many people think like, oh, animals do cellular respiration and plants do photosynthesis. Plants also do cellular respiration because remember, that plant isn't making glucose so that you'll come along and eat it later. That plant is making glucose so that hopefully, if the weather conditions go bad or it's cloudy for a long period of time, they can access that stored energy themselves. Comes in two stages. The first stage is glycolysis. It's anaerobic. It doesn't use oxygen. The second stage is respiration, and it does use oxygen. Glycolysis takes place in the cytoplasm, and respiration takes place in the mitochondria. The chemical equation for this is C6H12O6, glucose, plus 6O2, oxygen, goes to 6CO2, carbon dioxide, plus 6H2O, water. Well, where do you get the glucose from? You eat it, right? So you have your donut in the morning and you get glucose. Where do you get the oxygen from? That's the whole point of you guys breathing in. So you are breathing solely to get oxygen in order to do this process. And your heart is beating solely to move that oxygen around your body to where it's needed to get ATP. Carbon dioxide is a byproduct. Your body doesn't use it, but it's made in this process. And so you just exhale it. And water, same way, it's a byproduct. You're not using it, you just pee it out. Here it is in pictorial form. Big ungainly glucose molecule goes through glycolysis, which is anaerobic, doesn't require oxygen. It breaks this huge molecule up into smaller chunks. If you have oxygen, those smaller chunks go into the mitochondria and do Krebs cycle and electron transport. If you don't have oxygen, it comes down here and does fermentation, which gives you a little bit more energy. And then depending on what kind of organism you are, the byproducts are alcohol or lactic acid. So glycolysis, the first step, big glucose molecule goes in, and through a series of enzyme-driven reactions, like a chain link reaction, we take the big glucose and we break it up into two smaller pieces called pyruvic acid. As we're breaking it apart, we are generating energy because we're breaking bonds, right? And when we break the bonds, we release energy. When we talk about glycolysis, this is a chemical reaction. And like any reaction, you got to jumpstart it. You got to give it a little bit of energy. The way I like to think about this is if you're going to go make a fire, which is going to give off a lot of heat, a lot of energy, you have to add some fire in to begin. But that little match that you add in is nothing compared to the huge bonfire that you're going to make. So same thing with glycolysis. You put 2 ATP into it, but you get 4 out. Well, in the end, you really only gained 2. It's like if you were going to sell t-shirts on campus and you went to the wholesale factory and you bought them for $10 and then you turn around and sell them on campus for 20, your profit is only $10. Well, in terms of glycolysis, your ATP profit is only 2. Cellular respiration is what's going to take place if you have oxygen. It starts with pyruvic acid, that endpoint of glycolysis, and now you take that pyruvic acid and you put it into the mitochondria, and through a series of reactions, you get out a ton more ATP. So you get about 34 ATP at the end of this after you go through the whole oxygenated cellular respiration process. That's a huge gain. So 2 ATP if you don't have oxygen, 34 if you do. Obviously, as a huge organism, we want to be doing this cellular respiration with oxygen. The byproduct of it is carbon dioxide and water. Fermentation's what happens if you don't have oxygen. There are some organisms that live solely in this fermentation world, but they're microscopic. Think about yeast. Like when the pandemic hit, we all were baking, and so we were using yeast, and that's what you're going to use in your experiment later on this week. But yeast lives in the world of fermentation. When it does the fermentation, it starts with glucose, does glycolysis, and then does this fermentation kind of magic and squeezes out a little bit more energy. And the byproducts, if you're doing alcoholic fermentation, are going to be ethanol, which is an alcohol, and carbon dioxide. The carbon dioxide is what makes the bread rise. If you're a bacteria, you could do it with milk and do lactic acid fermentation and make cheese or yogurt, or with soybeans and make soy sauce. So lactic acid fermentation is what you guys are doing in times that you can't get oxygen everywhere in your body when you need it. So we're going to regenerate NAD so that glycolysis can continue, and there's the reaction. Think about when you might not be able to get oxygen to the appropriate cells. The one that comes to mind is if your coach is mad at you and is making you run sprints up and down the field. The first couple of sprints, you're breathing in, the oxygen moving through your heart rate and all of that are good enough to supply the muscles in your legs. But by the fourth or fifth time you're up and down that field, all of a sudden you're like, you are using ATP in those muscles faster than you can supply that area with oxygen to do cellular respiration. And so, as a last ditch effort, they do this lactic acid fermentation. The goal evolutionarily is that you would be being chased by like a mountain lion, and you can't just tell your coach, that's it, I'm done, I tap out and sit down. You got to keep running or you're not going to survive. This lactic acid fermentation pathway is there for those instances to keep you alive evolutionarily. Now it just makes you suffer a little bit with your coach. The lactic acid will actually build up around the muscles, and that's why when you're doing it, your muscles are going to burn a little bit. Once you're done with those sprints, the worst thing you can do is just sit down. You want to slowly be walking around, moving those muscles slowly, kind of trying to regulate your heart rate, regulate your breathing, bring that back down to normal. But you still want those muscles contracting, because that contracting of the muscles is going to bring oxygen back into that area, and the blood's going to take the lactic acid away from the muscles and take away the burn. Alcoholic fermentation occurs in yeast and other microorganisms. Same kind of thing as lactic acid. It's a way of staying alive when you don't have oxygen or you don't want to utilize oxygen. The byproduct here is alcohol and carbon dioxide. The carbon dioxide is what causes your bread to rise, and the alcohol is the same alcohol that we get for beer or wine. It is toxic to the system, and if it's not moved away from the yeast, it will kill the yeast, just like it's killing your brain cells. If you do have oxygen present, you're going to go through the Krebs cycle. So we have glycolysis, end up with pyruvic acid. Pyruvic acid, now with oxygen present in your system, will go to Krebs. Krebs is this circular cycle where we just keep popping carbons off of pyruvic acid. And remember, every time we break a bond, we get energy. At the end of this, we get ATP, which I could use right now, and FADH and NADH, which are carrying that hydrogen in bond form over to electron transport. This is occurring in the mitochondria, and it's where we get our carbon dioxide that you're breathing out. Electron transport is the end of this. We take the NADH and the FADH and we put it through the membrane of the mitochondria through all of these enzymes. And essentially what we're doing is we're moving the hydrogens through these enzymes, and we're going to accumulate them on one side of the membrane. But they don't want to be all cracked up like this on one side of the membrane. They want to be over here where they can spread out. And so they're going to go through facilitated diffusion through ATP synthase, which is an enzyme that acts like a generator. And as those hydrogens move through, it cranks out ATP. The hydrogens then bond with oxygen and make water as a byproduct. Your end goal on this is the 2 ATP that you gain from glycolysis and 34 ATP that you get in Krebs and electron cycle. This is only 38% of all of the energy that went into glucose. You only get 38% to make your muscles work. It's not that efficient. The rest of that energy is what's keeping you warm. It's the reason why you are a constant temperature, why you're that 97.8 or 98.7. That's the end of the lecture. Now, I went through that relatively fast, and you might want to hear it again. Awesome, this is recorded. But you also might want to hear it from a different voice. I recommend Khan Academy, Crash Course, or Bozeman Biology. I am a huge fan of Crash Course, which is why it's linked at the end of our lecture. And I do highly recommend that you go through and watch that. If you're someone who wants deeper detail, you're going to find it in this Crash Course. But remember that your test will only go to the detail of the lecture that you just watched.